Okay, so if I go back, the other thing we can do from the analysis result level, we can also export the results. This is a very simple export at the moment of just this, basically just this table content, but you can have that as a tab delimited text format, a CSV format, or an Excel format, which is probably the nicest into an Excel spreadsheet. And then you select your output file, you create a file on the file system, and you can output the data. Okay, so that is essentially it for the HLA haplotyping. It's a very simple function. The whole idea behind it is to keep it as simple as possible in order to do bulk haplotyping, basically. So if I go back up to the home part of the site, I can click on the data analysis side, and we can have a quick look around inside the data analysis part of the tool. Okay, so the first thing we see is the data analysis dashboard. So this half of the tool is more for general alignment of next generation sequencing short read data to reference sequences, and then we do a variant call and we can analyze the results in here, and we can start to filter the variants with various quality criteria to try and filter the list down to sort of a more interesting and smaller list of candidate variants that may have some kind of diagnostic effect that we'll be interested in. So the first thing, the way we recommend working with the data analysis dashboard is to actually add a profile. So we've created a number of pre-built profiles for the tool. So what the profiles include is a pre-configured set of reference data, so you don't have to manually import any reference data into the tool. It also includes a target definition. So one of the things that we have within this part of the tool, and the reason why the tool is called Omixon Target, is this concept of a target. We're going to have a look at that in a little bit later. So the profile basically contains reference data and a target definition. But I'll just import one now so you can see this in working. This is the BRCA profile. You can select a sequencer, Illumina, Roche, or Ion Torrent. So I'm going to select Illumina in here. Now, for demonstration purposes, I can actually run this import from a file, which is a little bit easier to do. So I'm just going to find my profile file. Where did I put this? There's not a test data. There we go. So, sorry, that's the wrong one. I selected the BRCA profile. So I'm going to run this from file. Normally this will download this file automatically from via the internet, and we'll install it into the tool without any other intervention. So this is running a little background task. You can see the status is running over here. It only takes a few seconds just to add a new profile. I'm just going to wait for that to finish. There we go, it's finished. So what this has done, actually, I can show you a little bit about what it's done in the background. So if I click on the references in here, this is HG19, which is what we use for our standard reference sequence. And what this has actually done in this particular run, it's imported this FASTA item here, which is for chromosome 17, which is what we tend to use for the BRCA sequencing. It's imported the DB-SMP, so the known variants for chromosome 17, and it's imported some background Illumina data and some sample data as well. So actually within this dashboard you can import your own reference sequences. So if you want to import a FASTA file, you can just manually do it in here. You can import some more DB-SMP data as well if you want to. So if I just go back to the data analysis dashboard at the top here. The other thing that the import profile has done is to create a new target, which is for BRCA1 and BRCA2, which includes some annotations for the various exons within BRCA1 and BRCA2. So this target will get used within the tool. You can configure this target within here if you want to. And I'll jump back to the data analysis dashboard. So this target will then get used within the tool in order to help you decide what is on target and what is off target. So on target for this particular target is defined as everything within the exons in that particular list. So adding a profile will also create you a first experiment for the profile and the first analysis. I think this is a good time just to quickly take a step back and talk a bit about the data management side within the tool. We have a small hierarchy for data management. It's just got 3 layers in it, really. So the top level of the hierarchy is the experiment. So this is an experiment that we've created by adding a profile called BRCA experiment. So if I click on that, it will show you a list of all the analyses within the experiment. And the reason why we've done this is so that we can basically share a design for an experiment across multiple analyses. So the idea here is that this will then make the processing for the various samples that you want to analyze a bit easier to do, because we've already pre-configured that we're working with Illumina, we're working with the BRCA target, we're working with human, HG19, and chromosome 17. This has already been configured at the experiment level, so we don't need to touch this again, basically, and the mapping and alignment tools will automatically pick up all of this experiment and design within here. So there's only one analysis within this experiment. I can create another analysis if I want to. So new analysis. This will inherit, again, as I say, all the configuration that we've already seen from the experiment, so I don't really need to do very much in here apart from just click through that wizard, and then I've got a new analysis. This one is empty. If I go back, this is the one that was created by adding the profile. So for this one, it's imported a little bit of example data. So if you want to have a play around with some example Illumina data, this is actually simulated data that we've generated that covers the BRCA1 gene. It only covers the BRCA1 locus. But so you can go in and work with this sample. So the second level of the hierarchy is the analysis, and this is where we group together the various samples that we want to work with. So there's a few more functions available within here. Again, you can go in and design the analysis. So if you wanted to have a slightly broader experiment, but then drill down to something a bit more specific on an analysis by analysis level. So for example, you might want to configure one target for BRCA1 and one target for BRCA2, in which case you can split the analysis level and have one analysis that is for BRCA1 and one is for BRCA2, just for example. So you can also do slightly more specific configuration within an analysis if you want to. We also have some bulk processing features at this level, at the analysis level. So you can import multiple samples. This will allow you to import multiple FASTQ files basically in one go, and again, if you want to, you can import paired data, and this will automatically create samples for you, one for each of the FASTQ or pair of FASTQ files that you choose in this particular wizard. So this is to help things with sort of bulk processing. The other bulk processing feature in here is that you can select all, you can use this function over here, select all, select all of the samples, and you can map and align and do a variant call basically for all selected samples with a single button, which is this one here. In this case we've only got one sample in this list, but if you had 100 you could do 100 in one go. But I'll talk a bit more about the map and align function when we look at the sample. So I'm going to click on the sample. You can either select the sample and go to the sample dashboard, or you can just click on the sample, which takes you to the sample dashboard. And in here we have all of the sample related functions. So in this particular case I've got some short read mapping data that's been done on an Illumina. As I say, this is actually simulated data, so we've just simulated this particular set. It's got within it some variants from the DBSMP that we've introduced into this data, so there's something to have a look at. And we've got some expected results. So in fact, all the variants that we've introduced into this data set, we've also imported here as an expected set of variant calls. So that once we've run the analysis, we can compare the expected results with the actual results automatically within the tool. We don't have to work just with raw sequencing data. We can also work with with data that's been mapped by any other tool in SAM or BAM format. We can import some Sanger data in here, so you can see that alongside your short read data in the genome browser that I'm going to demonstrate a bit later. And you can also just import variant calls. You don't have to import any kind of NGS data at all. So if you just want to compare variant calls across multiple samples, for example, you can just import them in here. The analysis options, once you've imported some NGS short read data in FASTQ format, you can then map and align that data, and you can demonstrate it. You can basically run through this wizard here, where it's automatically basically pre-configured within the tool. You don't actually have to choose any options within here unless you want to have some advanced parameters. You can just step through this, choose whether or not you want to do a variant call. It will automatically use the dbSMP function here, and then click finish, and it will run a map and align job on the system. Now, I'm not going to demonstrate this because it's going to slow my machine down a little bit too much. I'll probably lose the audio. So instead, what I'm going to do is I'm going to go back up and show you what I did earlier. So this is actually within the HLA side of the tool. So in here, I've imported already the HLA. I've added the HLA profile earlier on. So if I click into the default HLA experiment that's created by that process, HLA analysis, then in this case I chose to import some example data for both Illumina and Ion Torrent, and this is an Ion Torrent set. So this one has been run through the map and align process, so that's no longer available on the list. But what is available in the results section down here is we have the genome browser and the analyze sample variance function. So I'm going to click on the genome browser, and this takes us into the genome browser with a fairly high level view. So I'm going to maximize the view here just to make it a little bit easier to see what's going on. So our default genome browser view is done with a vertical display. So this is going to be the reference sequence track along here. This is the genes and the exons that we're working with for this particular target. The reference sequence is chromosome 6, which is where all the HLA genes are. And in this case, we've also got an expected set of results. So this is going to be displayed alongside the actual results in the genome browser automatically. Again, this particular data set is simulated data, simulated for the Ion Torrent machine. So as I zoom in to here, I'm just using the mouse wheel to zoom in. We can see the variants. Now some of them are outside the exon region, and some of them are inside exon regions. This is one of the exons here. This is HLA-A, exon 3. So as we zoom in, we can see a bit more detail. Once we zoom into this level of detail, we can start to see the short reads within the display. So this is an individual short read over here. This is showing the cigar for the short read. The little green blobs that you see, I can put on the mask mode. So the mask masks out everything except the differences between the reads and the reference. This is fairly standard for genome browsers. And because I've only got a single sample in here, I'm actually going to switch the view, and I'm going to rotate to the horizontal view. The vertical view is slightly more useful when you have multiple samples and you want to compare them side by side. But if I rotate the view, then this shows you the horizontal view. And this one's a bit easier for single sample visualization. So as we zoom in, we can zoom in over here, and we can see that on the actual variant track over here, it looks like we have a heterozygous mutation here. So the reference value is G. We're expecting an A variant in here, and we have actually found a heterozygous GA variation in here.